Hello, everyone. Welcome to TIDE's Oligonucleotide and Peptide Therapeutics Digital Week, brought to you by the producer of the face-to-face TIDES events. My name is Jonathan Royce, and I'll be the host for today's session. The topic today is there a better way to achieve peptide purity? But before we start, I'm going to cover some quick housekeeping items. If you experience difficulty with the audio or advancing slides, refresh your screen with F5. If you're experiencing other issues, hit the question mark button to receive assistance. At any time during the presentation, submit your questions into the Q&A window on the left-hand side of your screen. This is an interactive panel discussion, so we really encourage people to use that question and answer section in order to keep the conversation going. In 24 hours, you're going to receive a link to watch the recording of this session. You can also download a featured white paper in the resource list box on the right side of your screen. With that, I'd like to introduce our panelists, Elizabeth Denton from Biotage, Richard Johnson from Red Lead Discovery, and Jan Berluf from BioWorks, who's standing in today for Sanjay Pati, who unfortunately could not be with us. Sanjay is from Sun Pharma, but Jan will be taking his place today. We have prepared, I'll let you introduce yourselves, I think. So Elizabeth, why don't you start?
Hi, everyone. My name is Elizabeth Denton. As Jonathan mentioned, I have a PhD in chemical biology, and I've been working at Biotage for the last 5 years or so, primarily supporting the peptide space. So I work with customers in kind of all phases of their peptide discovery projects, primarily at the R&D, but we do kind of think about those processes further downstream as well. But not only synthesis, but also purification and evaporation, and really thinking about workflow efficiency and the best way to get from starting materials to final product most efficiently.
And Richard?
Yes. Hi, everyone. I'm Richard Johnson. I work at Red Lead Discovery, where I've been working for the last 7 years, and approximately 4.5 years in the peptide space, and I've been heading the peptide group for the last 3 years. So we are a CRO supporting preclinical mostly, but some farm dev support as well with the peptides.
And Jan?
Yeah, my name is Jan Berglof, and I work as a senior advisor in BioWorks. I'm one of the founders way back, and I have worked with purification basically all my life. After university, I started with the Pharmacia Biotech, and been head of applications research, and done a lot of other things around purification. So, but that's mainly been protein, but definitely peptides has been a growing part of that during the last years.
Thanks, Paul, for joining us today. We really appreciate you taking the time. We'll start by just going through some introductory slides to get the conversation going, and I'll lead you through these. So again, the topic today is about striking a good balance in achieving peptide purity and the techniques that we can use on the synthesis side, and also what we can do on the purification side. The therapeutic peptide space is really an exciting space. A bit like Jan, I've spent most of my career working with proteins and antibodies, but I've in the last year or so been really diving into this space, and it's incredibly exciting. There's a long history here. In 1923, the first therapeutic peptide was introduced, and that was, of course, insulin that was extracted from the pig pancreas. And since then, so much has happened on both the synthesis side and the purification side. In 1953, Professor Vignot described the first synthesis reactions related to oxytocin. And then in 1963, of course, Merrifield developed solid phase synthesis, which is a technique we still use today. And in 1984, received a Nobel Prize for that work. In 1982, the first recombinant therapeutic insulin was introduced to the market, and since then there's been an explosion of other therapeutic peptides that are primarily made through synthesis reactions. Today there are over 70 launched products on the market. In 2000, Roche in Colorado developed the first ton-scale reaction for producing therapeutic peptides. That was an HIV therapy that today is no longer on the market, but then was a breakthrough in the peptide space, a 46 amino acid peptide that was, again, produced at the multi-ton scale. And then today, as I said, we have over 200 clinical projects for therapeutics, and over 50 of them are approved today. If we look at the sort of state of the market, we can also see that this is a fast-growing market, growing faster than some of the other therapeutic entities on the market today. The market's expected to be, from a commercial standpoint, nearly $50 billion by 2025. It's growing nearly 10% per year, and the pipeline is very rich. So you can see in that upper left graph the therapeutic pipeline by clinical phase, and it's quite heavy in the early stages still today. And from the structural side, we see that the complexity of peptides is increasing. They're becoming longer, and the constructs, they're becoming longer and longer, and this is reflected in the data that we see coming out of the clinical pipeline as we see the average length of peptides increasing over time. Solid phase peptide synthesis, for me as a protein person, I think of this as the upstream of peptide production. It's a well understood reaction today, but it still has a tendency to sometimes produce impurities. So there can be peptides that have coupled incorrectly or been produced in a length that's shorter than expected. We can get mismatches along the way. And it's important that we try to control these as much as possible, but at the same time, we want to be able to produce these high complex peptides that, again, are growing in length and also in many cases growing in terms of complexity and also the commercial need. So this, the sheer volume of peptide that we're trying to produce. And then the downstream of peptides, very much dominated historically by reverse phase chromatography and still really the key technology that's being used to purify synthetic peptides. But also we see more and more people using ion exchange resins as an initial step to try to clean up some bulk impurities and protect those more expensive RPC resins downstream. And I think, you know, if we look at a sort of analogy to the world of antibodies, what we see is the same sort of dynamic that came into their world about a decade ago, when titers started to really rapidly increase on the upstream side. And at the same time, also impurities were increasing as well, creating a bigger burden for the downstream team. So there started to be a lot of discussion about linking upstream and downstream development teams, having them work closer to one another, working towards common goals. And this is just an article that the producers of this event, their parent company Informa, recently had on their website, which was about this sort of hidden danger, upstream intensification and the downstream mess you see there in the headline. And that's you know, an interesting analogy to the discussion that we're going to have today about these complex synthesis reactions that are becoming more and more complex, and how do we balance the need for higher complexity and higher production volume against the ability to purify these and to make something that can be used as a therapeutic substance. So again, just because the upstream is synthetic doesn't mean there are not impurities. There are lots of things that can be that one must consider. There are, of course, deletion sequences, truncation sequences, incompletely deprotected sequences, things that are modified during cleavage, and on other side reaction products that are also can be produced during the synthesis reaction. And then the question is, so how should we optimize, and what should we optimize for? Should we optimize for yield? Should we do it for purity? Is it the productivity of the process? And also, probably more and more, actually, we see people discussing about the environmental considerations around peptide production. So things like solvent consumption, and then what do we do with the solvent once it's been used in the reaction. So with that, I'll turn over to our panelists. If there's any initial discussions, or if you want to, or initial comments, or if you want to jump right into the questions, we can also do that. I'll go for a question then, I think. So
Yeah.
always a good way to get the discussion going. So let's start with this question that came in, which is about, you know, striking the balance. So what are you guys' thoughts about when is the right point in the process from going from discovery to lead development to process development? Where do you think that people should be putting their effort, the biggest effort, in to try to work out these protocols to provide good upstream reactions, but also to get purification protocols that are going to be workable in the downstream? What are your thoughts on that? Certainly, you can say that in the protein world, this is a discussion that's ongoing as well about, do you do brute force purification during discovery until you have a candidate, and then really work on the DSP, or do you put some bigger efforts in early on to make sure you have a process that will move into manufacturing quickly? What do you guys think?
Should I start?
Yeah,
it's a really hard question.
You're ready. Go for it.
We work in so we don't work with we work in the early phase. So we usually work in small scale, but the small scale we work in is from milligram up to, let's say, a couple of grams. And when we talk about milligrams, I don't think you need to do so much optimization to get out your milligrams. Sometimes you need to, depending on the sequence, but as soon as you come to a couple of grams, you can save so much time in optimizing the synthesis to get at least get the impurities that you get further away from your main peak. So if we I think in the lead development, then I would say that you should start really thinking about optimizing your synthesis for maybe not just peptide purity, because it needs to go a little bit hand in hand in time and yield, but also the impurity profile, because if you can get some impurities that is further away from your main peak, so the purification is easier, that is worth so much more than to gain a couple of percentage in yield with an impurity that is close.
I think the answer to that question kind of goes hand in hand with what Richard was saying, but also it really depends on where you're at in the process, right? So if you're still in early phases of screening libraries or lead development, you know, those experiments don't necessarily have to have as high of a purity. And in order to be most efficient, you've got potentially thousands of compounds you want to screen, right? And in that case, you really need to work pretty hard on synthesis to make sure that you actually have enough compound to run these screens on, and potentially not even do any kind of purification whatsoever. But then as that funnel kind of narrows, if you're thinking about both synthesis and purification simultaneously, all that does is make process easier, if that makes any sense. Because like Richard was saying, the impurity profile really makes the purification part the most difficult, and the bigger the column, the more expensive it is, the more solvent it's going to consume. And if you can kind of keep those impurities more distally removed from the main product peak, everything gets easier. But that's a synthesis optimization process. So I think it really kind of depends on where you're at. And just like Jonathan alluded to earlier, we're seeing a lot more early phase collaboration where the process group is getting involved, really with the discovery teams that are, you know, okay, this is the compound we think we're going to take. Let's think about these different strategies, because there are a lot of ways you can make compounds in the peptide space, whether they're semi-synthetic, where they only take a small portion that's chemically synthesized and express the rest of it from a purification perspective that could be more simplified. But when you're talking about the complexity of these compounds that are most attractive today, that can then become very difficult. So now you're back to solid phase synthesis type procedures to get these compounds developed.
Isn't it about productivity? In screening is one thing, and productivity in manufacturing is a totally different thing. And
Sure.
so that's really what guides the approach. So I don't think quick and dirty is probably not the right word. It's the productivity parameters are different. So when
Absolutely.
you scale up and really have your candidate, then of course cost of goods and other parameters like that become very important. So
Absolutely. Well, and just because the synthesis protocol worked really well on an automated synthesizer using whatever protocols you've optimized doesn't mean it's going to work on scale. When you go essentially back to more kind of manual, really large scale synthesizers, you can take some information from your automated synthesis protocols, but to some extent, my understanding is you kind of have to start back over when you go into really large scale syntheses.
the advantage, of course, the peptide industry has over protein is that you don't have to care too much about quaternary and tertiary structures and glycosylations and other things. So you in a way, the products are more easy to characterize, and therefore you can choose different approaches in another way than you can probably in the clean sort of real biotech.
Yeah, there's a comment here in the chat that controlling the impurities in synthesis is preferred compared to downstream, as the yield also improves when you convert the impurities. So the idea, I guess that's the idea, that in downstream, you're often making that trade-off between purity and yield. And if you can solve that in the upstream, you can sort of break that relationship to some extent.
But it's always a discussion. It's always a discussion. How much time should you spend on optimizing your synthesis to end up with this? And should you spend 2 weeks to optimize your synthesis to get a really nice, clean product for 5 milligrams that you could have done in overnight synthesis? So there's always the trade-off. And I mean, ideally, you should have 100% yield all pure after synthesis, but that's not how it is in real life. So.
I mean, that's actually one of the questions that's been asked here. One of the participants, one of the audience has asked, do you guys think that there is a future? Is there a space in the future where synthesis has become so good that downstream isn't needed anymore? Is that a realistic state to strive towards? That synthesis is a perfect reaction?
No. No, I would like to say yes, but I don't think so. There will always be something, and we have also to remember how things are. I mean, already today we can do things that are good enough for preclinical studies and screening crude off from the synthesis. But the other thing is too that what is approved in a drug today is the limits. What is approved is also getting lower. So as synthesis becomes more and more efficient, that we can get higher and higher crude purity, the impurities that is allowed in an API is also going down. So I think they will go hand in hand in the future as well. So when we can get out our 99% or 99.9% pure API from the synthesis, we have even harder limits from FDA and EMA to approve the drug.
Yeah.
Well, and I think kind of contrary to what Jan said, there is structure in peptides. And while it's to some extent almost the opposite effect of what you're looking for in proteins, oftentimes we're not looking for structure, but they can form aggregates and other tertiary structures that can really wreak havoc with everything else. So it's kind of the opposite conundrum, right? But at the end of the day, you still have to get rid of them. If you're and those aggregates, or if there's solubility issues that kind of prevent you from dealing with those kind of solutions and purification, I think those become limiting factors for synthesis as well. I mean, peptides will aggregate on the resin during synthesis, which even if you could make the reactivity profile 100% efficient, that structure is still going to mess with everything.
Yeah. There's a comment here in the chat that one of the audience has said here, I really like the approach to discuss with the discovery team, but that's not the reality. Usually it's too late when you need to develop a molecule from phase 1 or later. The upstream level must be optimized, but most of the times the upstream level is the key for purity and impurities control. What do you guys think about that comment? How do we bridge that gap between the discovery team and the team that's sort of the recipient of these processes? And do you agree? Do you agree that it's not the reality, or do you think that there are companies that are doing a good job of this, of linking their discovery team to their production teams?
I can't really answer that.
Mm-hmm.
I don't know.
But isn't it?
Go ahead, Jan. What do you think?
Yeah, isn't it sort of the conflict between call it the quick and dirty in the discovery and the need for cost of goods in production, and it tends to be starting too late. I think that my feeling that could many times be the case. You wait for this. You already have the compound. Why should I do anything about it? But then the realization that, well, we should probably have done that work a bit earlier comes later, so to say. So I think that's the conflict or the imbalance, if you call it.
Well, and I kind of agree with that, Jan, but at the same time, in the peptide space, the purification groups that I've worked with have less experience with handling peptides. So if you hand them a pile of junk and say, clean this up, I only need 2 mgs, they might really struggle with that. And so when you're talking about, especially in pharmas where they're handing off compounds to different groups, level of experience of handling these types of compounds really plays a big role. And if you can work really hard on synthesis, you may be able to avoid that altogether, but they also makes everyone's job easier downstream. So, I mean, the question, and I kind of agree with the statement that was made that it may not, I do know personally of groups that are trying to do this kind of negotiation process and early discovery with the process groups, but, you know, I can't speak to how well that has worked at this point for that perspective.
Let's switch gears a little bit, because there's a couple of questions here about hydrophobic peptides, and I'm just curious to hear if you guys have any experience with them. One of those questions here says, what strategies do you recommend for hydrophobic peptides? Even at the milligram scale, those can be difficult to isolate, even though the synthesis is relatively clean. You guys have any tips for working with hydrophobic peptides?
Yeah, it depends how hydrophobic. Yeah. But we have had some really hydrophobic that we purify them on normal phase, actually. So, but then they're really, really hydrophobic.
Yeah.
What was the underlying technology that you used? What, like a hydrophobic interaction resin, or?
No, so normal silica purification with
silicon. Yeah, hexane and ethyl acetate probably. But I guess the follow-up question is the purification so difficult because of solubility? And my guess is the answer to that question is yes. You can get the crude material dissolved in something like DMF or DMSO, but the second that you put it on a column and introduce it into an aqueous environment, it crashes out and wreaks havoc. So those are difficult to work with. Some areas that I've kind of thought about but never actually tried would be like a hillock type purification strategy, which may help with the solubility issues that you encounter when you do traditional reverse phase. I haven't tried it. We've kind of kicked it around a little bit internally to see if that might be an interesting option. But solubility becomes your biggest challenge, because if you can't get it to dissolve, you can't get it to interact with the stationary phase, and you're not going to achieve any purification.
We have had problems not only with very hydrophobic but also hydrophilic peptides that come in pure water. But there is ways to there is nice columns that can handle the pure water, and usually you can get some retention time and some cleaning up of them. And, there is some peptides we also, it's not the final purification, but you can get rid of a lot of impurities when they are hydrophobic by normal water organic extraction, and you can shake away a significant amount of impurities. But the final fine purification needs to be done on some form of column.
Yeah, and that's the big struggle, is you almost get confined to the parameters of the regulatory authorities, and recrystallizing a peptide is not the best course of action.
But I've never tried.
You've got to. I've never tried either, and I don't want to. But those are some of the issues that we get to deal with sometimes.
You mentioned pure water. Sorry, go ahead, Jan.
That's when you want to really spend the time on the optimizing the synthesis, so it's coming out really, really clean. And it could be a quick purification.
Yeah. Well, Richard, have you ever thought about kind of the opposite? I mean, most of the time we think about chromatography in general, and even to some extent in the protein space for Jonathan and Jan, is to catch the desired compound and dilute everything else off to some degree, whether it's specifically through a linear gradient like in reverse phase. But with really hydrophobic peptides, would you ever think about a strategy that basically prevents retention of the compound but retains the impurities? I don't know how you would do that, but it'd be you'd still have a column involved probably.
Yeah, that's what we call capture in protein chromatography. You have a capture step where you try to as selective as possible capture your target molecule. So, and of course, one could think about, of course, techniques that are used in protein chromatography. For example, if there is some histidines involved in the peptide, you could use metal ion, immobilized metal ion chromatography, for example, or something like that. So there are probably other techniques that has not sort of been considered. Normally, when we think about normal phase and we think about ion exchange, then you manipulate the salt basically. But the importance is also to manipulate the solvent content. We can see that purifying certain peptides in various amount of, for example, ethanol, and also acetonitrile and so on, that can greatly influence how the peak comes out and how it separates. So basically, with the mixing in solvents, you have more parameters to work with than you normally have. But maybe this business should look also at the protein people and the techniques they are using.
There's a question here about could we use hydrophobic protected a hydrophobic protecting group as a handle for purification. So it's sort of that idea of having a affinity resin, but this was really specifically during reverse phase chromatography. So could you use that hydrophobic handle could you essentially exploit it during reverse phase chromatography?
I think in the oligonucleotide area, there is published and presented that kind of approach, where they use an hydrophobic approach or RPC approach with the protective groups present, and then they split them off, and then they do a normal ion exchange, so to say, with the gradient to purify the rest. So that approach. So maybe sort of the peptide industry should look around a little bit at what other people are doing when they purify.
There's definitely work been done in that area. There's some recent technology that uses kind of effectively a chemical tag as an affinity handle to perform chemical affinity chromatography on synthetic peptides. There's also, I've seen something similar with a hydrophobic tag that facilitates solution phase peptide synthesis, more so than solid phase, is my understanding of the technology. And basically what that handle allows you to do is pseudo solid phase synthesis and crash out the product after your reaction is done, allow you to filter away those materials so you don't actually have to do a full chromatography phase between the individual chemical reactions that are performed when you're doing solution phase synthesis. So some of that kind of exists and certainly has applications within the peptide space, I think, especially in early discovery phases where the diversity of the compounds is a lot greater. When you get to phase one, you've kind of got to focus at that point and maybe a handful of compounds. And to some degree, I think it's important to focus on both because they feed into each other.
I mean, speaking about Jan's comments about looking at what protein scientists do, there's a question here about, we think that we'll one day be able to make more of the required peptides or most of the required peptides by recombinant techniques. Is single phase synthesis here to stay, or is recombinant technology a potential way forward here? What do you guys think about that? Something you've explored or seen people explore?
Yeah, well, the recombinant techniques will increase, but they will not replace. There's at least, as far as I know, there is a long development time to get a recombinant technique in working, so that you can produce your and if it is something that will be going into the clinical trials, maybe it starts getting interesting, and it's absolutely when it's a marketed drug. But in the discovery phase, you need to spend some to produce your different yeasts and bacteria to produce your peptides. I think it's not now. Maybe in 10, 20 years, we found a new way to produce bacteria and yeast, so it can go even in discovery, but not now.
I think the bigger issue is
in the protein world, people are talking about, in 10 to 20 years, there'll be cell-free production of protein. In the peptide world, there's a possibility that in 10 to 20 years, we'll be using recombinant techniques.
My feeling is that we would probably skip the recombinant techniques and go straight to cell-free, because the bigger issue in the peptide space is the use of non-natural amino acids. It's pretty pervasive, and most of that comes in to stabilize the compounds once they're in circulation. Peptides are notoriously unstable if you just let them be their native amino acid sequence. So there's a lot of work to incorporate non-natural amino acids, and those recombinant strategies to incorporate even one non-natural building block just aren't very efficient, let alone multiples of them. So that's a chemistry problem. And I think that my feeling is that's pretty much here to stay, the incorporation of all those building blocks.
Yeah.
What's your experience? There's another question here about lipopeptides that have no chromophore. Do you have any advice about those? This is another sort of class of these hydrophobic peptides. Any experience there?
Yeah, yes, a little bit. But we have worked with some with very little chromophores, and it usually works. There is, I mean, the amide bond has an absorption, very small, but it still has one. So it's possible to detect it on UV. But there's also other detectors that if that is a big thing you're working with, it might be worth investing in a refractive index detector or something else that could detect.
Yeah, certainly if you're doing a conjugation reaction where you're trying to separate non-conjugated peptide from product lipopeptide, distinguishing between those two by UV would be very difficult. But I guess my feeling is a mass detector is a pretty critical component in all phases of peptide characterization. And that would certainly be the easiest way to identify which one is which.
Yeah.
Let's talk a little bit about like selection of resins. So if we think a little bit about downstream, so in RPC, when do you go for these different types of columns, C4, C8, C18? What are the selection criteria that you use to try to sort of choose correctly from the beginning?
I mean, we mostly use C18 for 98% of the things that we use. I think we use C18. We have C8.
Would you call it a platform? Would you call it a platform then? C18 is your platform that you only deviate from when you have to?
Yeah. Yeah, I would say so. It's it works very well, and especially with the peptides. Most peptides, they work fine. We have worked with a couple of conjugation, protein conjugations and things like that, and then we switched over to C4, where we have a little bit better separation. But it's rare. To me, I think it needs to be. We need to be up in a good sized protein to start thinking about something else than C18.
Yeah. Or a different level of hydrophobicity. Like that's usually what also kind of drives it. Is we tried C18 and weird things happened. So now what else should we choose? I worked with a colleague that in graduate school that was making very hydrophobic peptides. And he actually ended up using a phenyl column to do most of his purification. So rather than a C18 functionality, there was a phenyl or a biphenyl functionality coming off of the sorbent, and seemed to have pretty decent results with that. But those are very high organic types of gradients that he was running. Like I think they were 70 to 90% acetonitrile or something like that, which certainly helps with the solubility problem. But his issue is he just couldn't get compound eluted off. It would like stick like glue to the sorbent. So then he had to find something that was okay with the solubility or lack thereof of a solubility profile of this compound, but also now I need to get it back too.
In that case, maybe he should have tried also different other hydrophobic ligands, because phenyl is quite strong. There is butyl, and there are other constructs too that could have been tried. But I think it sounds attractive to go that route.
The question becomes investment. So just like Richard was talking about, what's the return on a 2-week investment in synthesis? Well, HPLC columns are not cheap. And if you want to have I've never seen a sample kit of analytical HPLC columns that you could buy from the store for a relatively comparatively inexpensive cost where you could try all these different sorbents. So investing in something that you might use once and still doesn't work isn't super attractive to groups unless it has to be that.
Yeah. So we talked in the intro a little bit about the pressures now to try to reduce solvent consumption, and there's a question here about any tips on further downstream for reducing solvent use of purification and or removing solvent post-purification? Anything we can learn from the world of proteins or oligos? How much are you guys thinking about solvent use? Maybe at the scales you're at, it's not such a big issue yet, but.
No, but we do. I mean, I think everyone today, or most people today, want to reduce use of solvents and reduce their carbon footprint in some way. And of course, we think about it and try to optimize. And I mean, what we really try to do is if we look at HPLC and purification, we try to do very fast focus gradients. We work very much with step gradients to get down the purification time or gradient time. So, and also working with pretty big columns in general to get a lot of material onto the column and get it out. That's how we do it. I mean, if you think about the oligo world, at least when I was doing that many years ago, we were working a lot with ion exchange, and that is a water-based gradient, so that is good, and that we do to some extent with peptides as well. But usually we end up with some form of C18 step in the end with some acetyl nitrite.
Yeah. It's sort of the final C18 step.
Sorry.
I guess my feeling is a lot of times the C18 serves as kind of a desalting step. You can play around with ion exchange a little bit in early phases of purification, or maybe as a first pass cleanup, and then use your C18 to either change the modifier or your counter ion. Whether, you know, a lot of people like to use TFA to purify because it's a pretty strong counter ion and can help with resolution, but TFA is not very good for cells in your biological experiments. So then maybe use a final reverse phase step after you've cleaned up and actually use acetate as your modifier because it's a little more biologically compliant.
Solvents are probably the biggest driver of cost of goods on production scale. And of course, in order to minimize that, then comes also the resin type that you have, for example, very good capacity, dynamic capacity, so you can load a lot and still maintain a good selectivity when you elute the product. So that's one way. And of course, to have combined, let's say, an ion exchanger with which is an orthogonal technique to RPC, so to say, so you can maybe work with the first step with a lot of sort of water content, although you may have to mix in some solvents. And I think that's an approach you have to take in order to minimize the amount of solvents, because you can't avoid them, at least in modern time.
I think we go back to synthesis purification optimization, right? Like, if you work hard on synthesis, and even to Rickard's point, maybe you're not necessarily getting 99% purity off of the resin when you do synthesis, but if your impurity profile is well resolved, that enables you to do things like step gradients, maybe use alternative strategies that a lot of times is actually really difficult, because most of the times the impurities that you're seeing are single amino acid deletions, or they're very closely related, so they're very difficult to separate chromatographically. So taking advantage of those differences is the things you have to start thinking about from a chromatographic perspective. How are these compounds different? What can we take advantage of?
You guys there's a question here about polymeric RPC resins. Do you have any experience with that and what they can bring to purification? I guess this means non-silica based RPC resins.
Yeah.
I wouldn't say I have. Maybe someone else has.
I have done a little bit of work with at least polymeric shelled reverse phase columns. So still a silica core, but then a polymeric kind of decoration on it that the C18 is then, or the stationary phase is then conjugated to. They claim that they have higher stability and better and are more tolerant to a wider range. So the resins or the stationary phases last longer. They're more tolerant of huge pH swings, for example, without being damaged or degraded over the course of that use.
And the biggest advantage of polymeric is, of course, the resistance to sodium hydroxide and other cleaning agents. So that's a problem. Another problem can sometimes be the swelling and change of volume, which you don't want in a column, of course.
Well, we took easy questions so far. So now in the chat here that they want to ask a challenging question. So the question is about, this person works with tetrapalmitoylated peptides in clinical phase. And the question is, how would you purify them? Or do you have any idea? I hope I said that correctly. I had to write it down to make sure I said it correctly. Tetrapalmitoylated peptides.
Yeah.
That's outside.
Well, I guess the question is that's a lot of lipid, but the question is how much peptide is there in relation to all that lipid? If it's a 5 amino acid peptide and you've stuck 5 palmitoyl groups on it, you're probably it's gonna be a challenge. But if it's a 30 amino acid peptide, this is a different discussion.
Well, it was meant to be a challenging question, so you know.
Yeah.
It silenced the panel.
I assume they might have some problems now since the question came. No, I don't know, but or maybe just testing us on our knowledge.
Exactly.
So one thought is actually to use reverse phase. I have a colleague in the organization that actually separates lipids from each other. Different chain lengths, different saturation levels. And he uses reverse phase, but they're very strong gradients. You know, greater than 50% acetonitrile starting points. Oftentimes there are 80% acetonitrile kind of starting points using a C18. You're going to have to think really hard about your gradient. You may even go as far as an isocratic hold in reverse phase, which can actually work better than most people think it does for peptides. And it depends on your impurity profile, right? Are you trying to separate 3 palmitoyls from 4 palmitoyls? Because that's going to be a challenge too.
Yeah.
Richard has his thinking face on.
I don't know. Yeah, I mean, it's. I was just thinking, can we do this recombinant? No. Instead. No, but yeah. I mean, it usually. I think that if we would have a project like that here, I think we would go with start with C18 and see that would probably be where we will go and see does it come at any point during the 100% acetonitrile. Is there another solvent we should change to on C18 still? But yeah, I would go the same way there.
C4 might be an option just from a retention perspective. It's a little less retentive for those kinds of compounds, but yeah, that's a tough one.
Yeah. Good answers. Thanks for taking that challenging question.
I don't know if we help very much, but
I'm sure all of these things have been tried at this point in that compound's life cycle.
We touched a little bit earlier about process economy, and there's a question here about regenerating silica, and is there a possibility to regenerate silica as like new, or at least 90% of its original quality, to separate impurities and reduce column pressure, and to reduce the investment cost or operation costs? What are you guys' thoughts about that? What are your techniques for regenerating these RPC columns, and do you have any tips for users out there?
I mean, we wash them. We have a pre-column. It's a good thing to collect things that would never end the column anyway. So the pre-column is a good way to save it or make it work for longer. And then we wash them, and they can handle a lot of organic solvents for washing as well. Then you just have to do a new test injection, see how many plates you have, and see if it's okay, and then continue. But it is, I mean, the HPLC columns, even if they are expensive, it still is a form of consumable in the synthesis. So we need to think about it like that too. It should last a long time, but it's still a consumable.
Yeah. Well, and I think to Ricker's point, it's important to have a good QC process as part of that. So whether you've got a standard gradient, standard compound, standard mix to test your theoretical plates, the only way you're going to know if any of those procedures work is if you have a QC protocol. So that's got to be in place for sure. But I mean, if you've subjected your column to something that's actually going to cause C18 to leach off of it, high base concentrations, for example, there's not really any way to get that back.
Jan, I can almost anticipate your answer. You're going to say, protect that column with an ion exchange resin, right?
Good suggestion. Yeah, that's the point, of course, that you take out most of the impurities on the ion exchange resin first. So you have only small amounts that you have to take out on the RPC. We would call that polishing, where you fine-tune the purification. And then you get probably a longer lifetime and a better productivity. You can probably also load more on the RPC if it's more pure. So you save on solvents if we refer to that discussion. So absolutely, that's a good approach.
Yeah. We're starting
in general. Sorry. In general, I really like the two-way purification, or the two steps of purification and two orthogonal ways of purification. So instead of doing 2 runs on the C18 HPLC with acetonitrile water, it's only small change to do one with methanol water and then with acetonitrile water can make a huge difference, because some impurities separate much better in methanol water system than they do in acetonitrile water. So two steps like that, or an ion exchange, or something else, an orthogonal purification in combination is really a good way to go for purification.
I've had similar experiences with pH shifts, depending on where your isoelectric point of your peptide is.
Yep.
Run a first pass in a pH 10 kind of acetonitrile water, then a second pass with acidic modifiers. You just have to be careful with those with your instrumentation, though, because not all instruments or columns are compatible with all those kinds of things. So before you try, or if you're switching the same instrument between two modes like that, ask your manufacturer first, because you can break things that way.
Yeah.
Coming from an instrument supplier company, you can break things that way.
Well, we are starting to reach the top of the hour here. Do you guys have any sort of closing thoughts or advice for people about this question around synthesis and purification and striking the right balance? I mean, we've had a great discussion here today, and I think we've already covered lots of things. But just any sort of closing words of wisdom you want to leave with the audience? Don't be bashful.
Be brave. There's a lot of, I guess I have found in working with different groups that there's a lot of, we do it this way because that's the way we've always done it. And there are new developments that are coming through and alternative strategies, both in synthesis and on the purification side, that maybe not be super common yet or aren't necessarily talked about because they're buried in process literature or they're used in different areas. But be brave and try something different. You might be surprised at how well it works. You also might be surprised how bad it works, but you can at least, learn something from it and maybe improve your strategy.
Yeah, it's probably less known in the sort of this industry that ion exchanges could actually do a good work. And because all the way from screening and so on onwards, RPC has been the main tool. So that means if you have a hammer, all problems then becomes a nail. And I think that changing also this capture and first purification using ion exchange, also together with various solvents, can maybe solve a lot of problems on the RPC side.
Yeah, I agree with previous speakers, but don't forget to improve your synthesis as well. I can add then. No, but depending on the synthesis, it could be good to what scale and how many times, or is it a series of compounds, similar ones? And it might be good to run one synthesis, a little bit look at what it is, or identify problematic couplings before, give them some extra attention in the synthesis just to get it up, and then absolutely look at 2 orthogonal purifications. I like ion exchange chromatography when you can use it. I think it's a great tool, and then some polishing up with a C18, or I think that's a good way to go forward.
Great. Well, Elizabeth, Richard, Jan, thanks for this discussion. I learned a lot today. I hope the audience did as well. Just a reminder that in 24 hours, you'll all receive a link to watch a recording of this session, and you'll be able to download a featured white paper in the resource list box on the right-hand side of your screen right now if you're live, and you can presumably do that after as well if you're going to watch an on-demand version of this talk. So thank you again very much for taking valuable time out of your days. Really appreciate it, and I hope that the audience enjoys the rest of this exciting week around oligos and peptide therapeutics.